So Badrilla is a small life science antibody manufacturer in the UK. We make polyclonal antibodies, some monoclonal antibodies to cardiac targets in the main. But we're also interested in developing tools that allow people to use those sorts of reagents, and in fact any antibody reagents, in a genuinely quantitative setting, and that's what I want to talk to you about today. So we've been collaborating with a number of university groups in the UK to achieve this. And so I'll introduce you to the tools to look at the analytical performance of your antibody-based test, both as a manufacturer perhaps, and certainly as an end user. The people who've done the work, are largely a mass spectrometry team led by Rob Bainan in Liverpool and a case PhD student between the company and Liverpool, Richard Bennett. So this is the type of molecule we're going to be talking to you about. It's a calibration standard. And in simple terms, it's a collection of epitopes to the series of antibodies to the markers you're trying to follow, concatenated into a protein scaffold with desirable features for whatever the assay is that you're trying to do. Or it's a scaffold which can be, after purification, derivatized covalently with the entity, the epitope that you're trying to study. So two incarnations of this type of material. And we're going to be using it as calibration materials. We can then look at assay analytics, analytical performance, and we can get our measurements all the way down to a completely stable chemical event, which is the number of copies of the target you're measuring per cell in your biological specimen. And we're hoping it's going to help us all standardize the workflows that we're performing. So that's what I'm going to cover. So we're here because of this irreproducibility problem, but this is the abstract from an article a couple of years ago by Anne Plant and her colleagues, and she is at the US National Institute of Standards and Technology, and in her view, irreproducibility is only part of the problem of metrology that research faces. And the other parts of, and it's an important part, which I'm not going to talk to directly, but the other parts are that experiments must have the appropriate controls, reference materials, statistics applies, and informatics for it to be a complete, robust package of work. And in some respects, Arthur was talking about that in sort of the immunohistochemistry guise yesterday. So research is going to be needed, she says, to establish the tools for such a set of measurements, which is what we're covering. So we've been at this for 10 years, and have been developing these calibration tools, which we hope will help validate the antibodies in catalogues. It will allow the experimentalists to conduct well-constructed work, where they can, really look at the technical performance of their work, incorporating quality control standards, incorporating calibration standards, and perform the right analyses. So we started thinking about this sphere, the end user, and these calibration standards helping both the execution of an experiment and the analysis. But for this audience, perhaps we're interested in what can it also do for the antibody producers to help them understand the quality of the antibodies they've got. So most of the applications that we've studied so far with our own antibodies and with others are Western blot-based, and this is about a half of all immunoassay work in the research field. There are about 250,000 papers in the literature with Western blotting, sort of as a tag to find, and there are about 20 that have been properly calibrated with a calibration standard. This is one of about 10 years ago, nothing to do with ourselves, where they incorporated a full recombinant protein for their target in different amounts, in different lanes, we can see here, and then they put BSA in so that the protein loading was identical lane to lane. That's a Kumasi stain up here, and then they're essentially performing Western blots with different amounts of their recombinant protein. So it's been done by many people using full-length reference materials. And as I'm going to show with our approach, that brings us the ability to come all the way down to how many copies of the molecule am I measuring per cell. The trouble with that approach, which we're hoping to get past, is that the full-length molecule of the standard looks exactly the same as the full-length molecule of your sample, and Western blotting isn't a very high-throughput approach anyway, sort of which poses multiplexing problems. So our design allows us to put in information to pull the epitope calibrant away from the native marker and bring multiplexing. And we can also concatenate them, as I'm going to show you, and make one reference material serving a number of different assays. So the first study that I want to just share with you is how we went about checking whether or not this was a viable approach, even though there are 20 episodes in the literature which offer confirmation it is viable in other people's hands. And so we matched up our approach with quantitative mass spectrometry. So if we think about a protein, it has got an epitope, and for the mass spec, we can take individual tryptic peptides, which have previously been shown to fly in a mass spectrometer as standards for mass spec. The same is true for each of the proteins in our learning set, and then we construct a synthetic protein that contains epitope information for target A and tryptic peptides for target A, sort of a very simple approach. That type of material can then be manufactured as a gene, expressed in E. coli with heavy forms of lysine and arginine, such that the cleavage for mass spectrometry results in a heavy peptide. And that heavy peptide from the standard can be easily spotted compared to the not heavy peptide from the biological material. So here's the workflow. We take our synthetic calibration material that I've just described, we dope it into the biological samples in a known amount, and then we process those samples either for mass spectrometry, which involves tryptic digestion, or with no digestion at all for Western blotting. And we've chosen to use an automated Western blot platform. Manufacturers are actually here because the data quality is very high with that platform. And we're wanting to know whether or not we get equivalent analytical performance out of the two tests. So this is what the mass spec data looks like. In blue, we've got the heavy peptide for one of the markers, and in red, we've got the biological form of that peptide. By getting an integral of the peak area and knowing how much of the heavy material was introduced, all of which is in our control, we can simply, from the ratio of those two, work out the quantity of the biological source of that peptide. And in this training set, which is components from the NF-kappa B pathway, we see that in the presence or absence of an agonist, TNF-alpha, we get a very big change in the expression level of this particular protein. So we can see the red now becomes extremely large when TNF-alpha is present. If we look at the Western blotting, sort of it looks like I showed on the previous slide from other groups. Here's our calibration material in known amounts in a serial dilution, and that very pale signal is the biomarker in the absence of TNF-alpha. When we come to the circumstances when cells have been exposed to TNF-alpha, up goes the level of expression of this marker, this protein, and again we can use the calibration standard to quantify it. So this is ECL-based Western blotting on this platform, and for the markers of this study, they all had a very linear relationship between the signal strength and the amount of the calibration material over about 3 orders of magnitude of loading concentration. And here is the comparison between the markers measured by mass spectrometry, which is in orange, and measured by Western blotting in blue. We had 5 in the series. Mass spec actually only found 3, which was disappointing. Western blot found 4, but where the 3 that were comparable or were spotted by both are each measuring very similar quantities of the marker in the same specimen. And also the standard error of the measurements is very similar between Western blotting and mass spectrometry. If we look at plus and minus agonists, again, the 2 techniques report it identically. So in essence, that shows us that there is promise in the technique. It can be used in this way. How about the other configuration where we're trying to look at post-translational modifications? Here we take a scaffold with a single site that is chemically reactive. It's a dihydroalanine in this case. So it was previously a cysteine. It's been modified chemically to make a dihydroalanine. And then it will be attacked by a thiol on a peptide, which is a phosphopeptide, to create this sort of a displayed epitope. We can follow that by mass spectrometry, so clearly the additional mass of this peptide is very, very easily spotted in the mass spec. And then we can compare, by Western blotting again, the ability of an antibody to see the dephosphorylated form of this, which is this lane, and the phosphorylated form, which is this lane. So an antibody that we didn't know was phosphospecific, or rather phospho-upset in this case, clearly is. It doesn't like that serine residue being phosphorylated, whereas one which has been generated to be phosphospecific is exactly that. So it's very straightforward to manufacture and display epitopes to a variety of peptide and modified peptide haptens. The last thing I wanted to do is show you sort of another piece of work where we're looking at a comparison of different antibodies on the market for a project. So we have a project where we're interested in kidney forms of a molecule called NGAL. You know, what's interesting about NGAL, it might be a diagnostic marker for certain forms of injury, which is the direction we're going. And the molecule is a monomer, a disulfide-linked homodimer, and actually disulfide-linked heterodimers. We then took 3 antibodies from different vendors. They're all monoclonal antibodies on this automated Western platform. And only 2 of them were able to see the monomer. The third one failed to detect it in this guise. But we can then follow up and establish the dynamic range of detection of that molecule. It's not 3 orders of magnitude in this case, you know, sort of. So we can't take our view of one marker and one assay and apply it to any other. We've got a very limited dynamic range. This is only half a log unit of active measurement range. When we then have a different sample, so E. coli, when they express this protein, actually predominantly express the dimer and very little monomer. Now we get, again, the same 2 monoclonal antibodies from vendors 1 and 2 can detect that. Vendor 3, the antibody just wouldn't detect it at all. And when we come to the quantitation, again, it's a very steep relationship, and it's shifted. You know, sort of, this is half a log unit to the right of this. So here are antibodies. It's the same protein. One is a monomer, one is a homodimer, and yet the antibodies are able to bind with different affinity to those different species. That can only be determined by very careful consideration of standards of this type. So, in my view, this allows us to really get to know the qualities of the assay that our antibody achieves, and it would allow us, if we wanted to compare, as a manufacturer, the performance of one batch of this antibody to the next one that's been prepared, etc., to demonstrate to our satisfaction that it is equivalent batch to batch. So I'm going to conclude there. Calibration standards may prove to be an important accessory to the antibodies we're using, primarily for the end users, so that they can be measuring things robustly, getting all the way down to how many molecules per cell am I measuring. The approach that we've developed can be used for proteins as long as the epitope is described. You know, sort of, and that's about half of all antibodies in the commercial world. The epitope is known, not always publicly disclosed, but it is known by the manufacturer. It can be used for all of PTMs, although we've only actually shown phospho-PTMs. We can do single standards, multiple standards, and obviously multiplex it in all cases with the biological specimen. The assay characteristics that this allows us to determine is the specificity, i.e. phospho versus not phospho, or whatever is the question. The sensitivity, so we could really tell you whether or not the assay or the antibody is capable of measuring down to this particular limit of detection what the dynamic range of measurement is, and as I've already said, whether or not we can achieve batch-to-batch consistency in our preparations. Thanks very much.
So any questions?
So I had a question about the previous slide and just the kind of lack of a dose response.
Yeah.
Antibody 1, and do you think that's because of the enzymatic detection you're using, or?
So antibody 1 actually is the antibody that we've taken forward. It has got a dose response. It's very sharp. It is enzymatic, but so too were the earlier slides. Sort of it's exactly the same modality. So I think we've just got a very abrupt, transition from the lower limit of detection to the top.
And do you think it's really that low affinity that you haven't reached saturation yet, maybe 5 micrograms?
Yeah, so I don't know. I haven't gone to, sort of higher and higher to know whether, is that the true plateau? That's just a 4-point logistical fit.
Hi, I just had a question about your earlier slide where you mentioned that you got 3 out of 5 positive results by your SRM mass spec assay. Could you just comment on why that might be, that you weren't able to detect?
To do 5 out of 5.
2?
So the way that we designed, wherever we could, we used empirically observed tryptic peptides, and therefore they were highly reliable. But that didn't cover all 5. So for some, we had to use algorithms that would predict whether or not there would be clean cleavage, no post-translational modification within the product, and it is likely to fly. We didn't get it right. Thank you.